alters binding protein signal sequence with DNA encoding factor X. Factor X2. And now recognition site as a linker peptide separating the two genes. When the genetic fusion on a plasmid vector was used to transform E. coli cells as expected, a large fraction of the fusion protein was found to be localized in the host cell periplasm. Functional interleukin 2 could then be released when the fusion protein by digestion with factor X. In many instances, when foreign proteins engineered for secretion are overproduced in E. coli, the precursor form is only partially processed, with about half of the secreted proteins retaining the leader peptide and the other half being fully processed to the mature form. This is probably the result of overloading some of the components involved in the secretion process. If this is the case, then it might be possible to increase the ratio of processed to unprocessed proteins by increasing the level of expression of some of the limiting components of the protein secretion pathway. This hypothesis was tested in a series of experiments in which a plasmid containing both PRLA4 and SEC-E genes, which encode major components of the molecular apparatus that physically moves proteins across the membrane, was introduced into E. coli host cells. Following this augmentation of the host cell secretory machinery, the fraction of the clone protein, in this case, the cytokine interleukin 6, that was secreted to the periplasm as the processed form with the signal peptide removed, increased from 50% to more than 90%. In many cases, secretion of heterogeneous or heterologous proteins in E. coli is dependent on the translational level of the protein, and the foreign proteins that were translated most efficiently were not necessarily secreted to the greatest extent. Sometimes too high a level of translation of a foreign protein can overload the cell secretion machinery and inhibit the secretion of the protein. Thus, one way to ensure the secretion of a target protein occurs most efficiently may be to lower the level of expression of that protein. Next is secretion into the medium. So before that, let me just show you the diagram, a schematic representation. This is the representation. Okay, take a screenshot if you can. Schematic representation of protein secretion. The ribosome is attached to a cellular membrane, and the signal sequence leader peptide at the end terminus is transported by the secretion apparatus across the cytoplasmic membrane, following by the rest of the amino acids that constitute the mature protein or peptide. Once a signal peptide has crossed the membrane, it is cleaved by an enzyme called a signal peptidase. Associated with the membrane proteins as well as secreted proteins generally will contain a signal peptide prior to its removal by processing. This lecture would be concluding in a minute from now, and the additional portion, the ensuing portions, will go into the third lecture. Please stay tuned and make notes. Please do not commercialize any portions of my lecture. Please use it to the best of your preparations for the concerned exams and to bettering or augmenting your career. Best wishes. Special note that these lectures are being spelled out word by word and with special significance to those who are visually impaired as well, and to all those students, and each and every student in distress or having any inconvenience to access such notes. Let's thank the authors of this entire text and preparation. We thank Glick, Bernard, Jack, and Cheryl for their continued support through their book Molecular Biotechnology, Edition 4. Thank you, and we continue with the subject and to going into the next third lecture shortly. Secretion into the medium. E. coli and other gram-negative microorganisms generally cannot secrete proteins into the surrounding medium because of the presence of an outer membrane that restricts this process. There are at least two solutions to this problem. The first is to use a host organism gram-positive prokaryotes or eukaryotic cells, both of which lack an outer membrane and therefore can secrete proteins directly into the medium. The second solution entails the use of genetic manipulation to engineer gram-negative bacteria that can secrete proteins directly into the growth medium. In general, relatively few proteins pass through the outer membrane of E. coli. However, some gram-negative bacteria can secrete a bactericidal protein called a bacteriocin into the medium. A cascade mechanism is responsible for this specific secretion. A bacteriocin release protein activates phospholipase A, which is present in the bacterial inner membrane and cleaves membranes, phospholipids, so that both the inner and outer membranes are permeabilized. Some cytoplasmic and periplasmic proteins are released into the culture medium. Thus, by putting the bacteriocin release protein gene onto a plasmid under the control of a strong regulatable promoter, E. coli cells may be permeabilized at will. E. coli cells that carry the bacteriocin release protein gene are transformed with another plasmid carrying a clone gene that has been fused to a secretion signal peptide sequence. The clone gene is placed under the same transcriptional regulatory control as the bacteriocin released protein gene so that the two genes can be induced simultaneously with the target protein being secreted into the medium. Although secretions of E. coli proteins into the growth medium is quite rare, the small protein YEBF is naturally secreted into the medium without lysing the cells or permeabilizing these membranes and various proteins. And continuing on figure 6.3, E. coli cells engineered to secrete a foreign protein to the periplasm by fusing the gene of interest green to a secretion signal A and to growth medium by permeabilizing cell membranes with a bacteriocin release protein encoded on another plasmid red B. This is take a screenshot and make the best use of it. And they are fused to the C-terminal end of YEBF following the removal of the signal peptide. The anti-fusion construct is secreted to the medium. It is believed that the pre-YEBF foreign protein fusion is first secreted across the E. coli cytoplasmic membrane to the periplasm with the concomitant removal of the signal peptide. Next, the leaderless YEBF foreign protein fusion is secreted from the periplasm to the medium by an unknown process that involves specific amino acids that are a part of YEBF that interact with an unidentified receptor on the E. coli outer membrane. To date, researchers have reported facilitating the secretion to the medium of human interleukin-2 A 15K Dalton's hydrophobic protein. And bacterial alpha amylase, a 48 kilodalton hydrophilic protein, and alkaline phosphatase 94 kilodaltons, demonstrating that a wide range of proteins may be secreted to the medium using this system. The next step in the development of the system will likely involve engineering a readily cleavable linker region between YEBF and the protein of interest so that the protein of interest can be recovered in its native form. In addition, this approach should make it easier to avoid contaminating purified recombinant proteins with E. coli liposaccharides, lipopolysaccharide, which is a pyrogenic toxin that may be released upon lysis of the bacterial outer membrane and is therefore a serious concern when the proteins are produced by use as therapeutic agents. YEBF is not the only E. coli protein that is secreted into the growth medium. The flagellate type 3 secretion apparatus secretes a protein flagellin to the growth medium. Thus, it is possible to fuse a 173 base pair translated DNA fragment upstream of the gene encoding flagellin as well as transcriptional terminator from the same gene to a gene encoding a protein of interest, with the result that the protein of interest is efficiently secreted into the medium. However, this system is not applicable to all proteins. Have a look at the figure 6.31, secretion following expression in E. coli of YEBF interleukin 2 fusion proteins into the growth medium. The protein synthesized in the cytoplasm includes a signal peptide yellow, which is shown. Let's go down and have a look. Yeah, that's there. That is excised when the fusion protein is secreted to the periplasm. The YEBF interleukin 2 fusion protein is then secreted from the periplasm across the outer membrane, so to the growth medium. YEBF is shown in blue and interleukin 2 in green. This is the diagram here. We follow on to the next topic, the metabolic load. The introduction and expression of foreign DNA in a host organism often changes the metabolism of the organism in ways that may impair normal cellular functioning. This phenomenon, which is multifaceted biological response, is due to a metabolic load, metabolic burden, metabolic drain that is imposed upon the host by the foreign DNA. A metabolic load can occur as a result of a variety of conditions, including the following. A1. An increasing plasmid copy number and or size requires increasing amounts of cellular energy for plasmid replication and maintenance. 2. The limited amount of dissolved oxygen in the growth medium is often insufficient for both host cell metabolism and plasmid maintenance and expression. Overproduction of both target and marker proteins may deplete the pools of certain amino acids, tRNAs, or even certain amino acids and or drain the host cell of its energy in the form of ATP or GTP. When a foreign protein is overexpressed and then exported from the cytoplasm to the cell membrane, the periplasm, or the external medium, it may jam export sites, thereby prevent the improper localization of other essential host cell proteins. Host cells with unusual metabolic features such as naturally higher rate of respiration, example Azotobacter spp, are more likely to be affected by these perturbations than are other host cells. The foreign protein per se may interfere with the functioning of the host cell, for example, by converting an important and needed metabolite intermediate into a compound that is irrelevant or even toxic to the cell. This is figure 6.32, which is a schematic representation of the biological consequences of a host cell of overexpressing a foreign protein and generating a metabolic load. When there is no metabolic load, the cell has access to sufficient energy and resources. The overexpression of a foreign protein, shown here as being analogous to these cells wearing a heavy backpack, prevents the cell from obtaining sufficient energy and resources for its growth and metabolism, so that it is less able to grow rapidly and attain a high density. Sufficient resources and energy, insufficient resources and energy. So one of the most commonly observed consequences of a metabolic load is a decrease in the rate of cell growth after the introduction of foreign DNA. Sometimes a metabolic load may result in plasmid-containing cells losing a portion of the plasmid DNA. Even in the presence of selective pressure, all or part of the recombinant gene may be deleted from the plasmid, since cells growing in the presence of a metabolic load generally have a decreased level of energy for cellular functions. Energy-intensive metabolic processes such as nitrogen fixation and protein synthesis are invariably adversely affected by a metabolic load. A metabolic load may also lead to changes in the host cell size and shape and to increases in the amount of extracellular polysaccharide produced by the bacterial host cell. This extracellular carbohydrate causes the cells to stick together, making harvesting, example, by cross-flow microfiltration procedures and protein purification more difficult. Well, secretion into the medium. E. coli and other gram-negative microorganisms generally cannot secrete proteins into the surrounding medium because of the presence of an outer membrane that restricts this process. And these are at least two solutions to this problem. There are at least two solutions to this problem. The first is to use as host organisms gram-positive prokaryotes or eukaryotic cells, both of which lack an outer membrane and therefore can secrete proteins directly into the medium. The second solution entails the use of genetic manipulation to engineer gram-negative bacteria that can secrete proteins directly into the growth medium. In general, relatively few proteins pass through the outer membrane of E. coli. However, some gram-negative bacteria can secrete a bactericidal protein called a bacteriocin into the medium. A cascade mechanism is responsible for this specific secretion. A bacteriocin can release protein activates phospholipase A, which is present in the bacterial inner membrane and cleaves membrane phospholipids so that both inner and outer membranes are permeabilized. And now the metabolic load. The introduction of an expression of foreign DNA in a host organism often change the metabolism of the organism in ways that may impair normal cellular function and functioning, with the following that have been enumerated. Translational errors occur in growing E. coli cells at a rate of 2 into 10 raised to minus 4 to 2 into 10 raised to minus 3 errors per cell generation. However, when a particular amino acid tRNA becomes limiting, as is often the case when a foreign protein is overexpressed in E. coli, there is an increased probability that an incorrect amino acid will be inserted in place of the limiting amino acids. This frequency of errors diminishes the usefulness of the protein as a therapeutic agent, since 1. The specific activity and stability of the target protein are significantly lowered, and 2. The incorrect amino acids may cause the protein to be immunogenic in humans. A well-designed experiment can minimize the impact of the metabolic load, optimize the yield of the recombinant protein, and enhance the stability of the transformed host cell. For example, the extent of the metabolic load can be reduced by using a low copy number rather than a high copy number plasmid vector. An even better strategy might be to avoid the use of plasmid vectors altogether and introduce foreign DNA directly into the chromosomal DNA of the host organism. In this case, plasmid instability will not be a problem with an integrated cloned gene. Without the plasmid vector, the transformed host cell will not waste its resources synthesizing unwanted and unneeded antibiotic resistance. Effective plasmid copy number on host cell growth rate. We see E. coli HB 101 with plasmid copy number, relative specific growth rate. The marker gene products. Chromosome integration is particularly important when a genetically engineered organism is destined to be released directly into the environment. The use of strong but regulatory promoters is also an effective means of reducing the metabolic load. In this case, the fermentation process is performed in 2 stages. During the first or growth stage, the promoter controlling the transcription of the target gene is turned off, while during the second or induction stage, the promoter is turned on. When the codon usage of the foreign gene is different from the codon usage of the host organism. Depletion of specific amino acid tRNA pools may be avoided by either completing or partially synthesizing the target gene to better reflect the codon usage at the host organism. However, since this is not a simple procedure, such an approach is likely to be used in only a limited number of instances. Nevertheless, in one study, it was found that levels of the protein streptavidin were 10-fold higher in E. coli when expression was directed by a synthetic gene with a G+C content of 54% than when it was directed by the natural gene with a G+C content of 69%. Although it may at first seem counterintuitive, one way to increase the amount of foreign protein produced during the fermentation process is to accept a modest level of foreign gene expression, perhaps 5% of the total cell protein, and instead focus on attaining a high host cell density. An organism with a 5% foreign protein expression level and a low level of metabolic load that can be grown to a density of 40 grams dry weight per little produces more of the largest target protein than one with a 15% expression level for the same protein and a cell density of only 5 to 10 grams dry weight per liter. Okay, fine. Now we are on to the summary of the entire chapter 6. The production of a protein requires that the gene be properly transcribed, and then that the mRNA be translated. In prokaryotes, a promoter region is necessary for the initiation of transcription at the correct nucleotide site, and a sequence at the end of the gene, a terminator, is essential for the cessation of transcription. Clone genes often lack these signals. Consequently, for expression of a clone gene in a prokaryotic host cell, the appropriate signals recognized by the host must be provided in the correct locations. Moreover, the aim of many biotechnology applications is to produce large amounts of protein, so it is necessary to use a promoter that supports transcription at a higher level, strong promoter, and that is compatible with the RNA polymerase of the host cell. However, continuous transcription of a clone gene drains the energy reserves of the host cell, so it is necessary to use a promoter system whose activity can be regulated either by the addition of a low molecular weight compound or by changing the growth temperature. In addition to utilizing promoters in their native forms, it is possible to alter the DNA to produce promoters with a wide range of activities. Efficient protein synthesis from a gene depends on specific sequences in its mRNA, and often for clone genes, other manipulations are required to ensure the stability of the protein and, if necessary, its secretion. As part of the gene engineering process, a ribosome binding site is placed in the DNA segment that precedes the translation initiation site, start codon, which also may need to be added. Finally, a termination sequence may be placed at the end of the clone gene to ensure that the translation stops at the correct amino acid. If secretion of the protein is desired, the DNA sequence preceding the clone gene should include a signal sequence in the same reading frame as the target gene. Lack of stability, of the protein that is encoded by a clone gene is another complication that is often encountered. For example, the recombinant protein can be degraded by a proteolytic enzyme to the host cell. One strategy to overcome this problem is to alter the clone gene so that it encodes one or more additional amino acids at its N-terminus. In this form, the recombinant protein is no longer rapidly degraded. In addition, the amino acids that are added to the recombinant protein can sometimes be used for purifying the fusion protein by, for example, immunoaffinity column chromatography. In these cases, the junction point of a fusion protein is usually designed to be cleaved in vitro, either chemically or enzymatically. Fusion proteins may also be used to facilitate the purification of the target protein. Most microorganisms that are used to express foreign protein requires oxygen for growth. However, oxygen has only one limited solubility in water and is rapidly depleted from the growth media of actively growing cultures, especially when the cultures attain a high cell density. Attempts to deal with the limited amount of dissolved oxygen available for cell growth and maintenance have included 1. Utilizing microbial host strains deficient and the production of some proteolytic enzymes that are produced during the stationary phase, and 2. Introducing the gene for the vitriocellular SB hemoglobin that binds oxygen from the environment, creating a higher level of intracellular oxygen and thereby causing increases in both host and foreign protein synthesis. By increasing the numbers of copies of the clone gene, the production of the protein is increased, and under these conditions, the stability of the product is often enhanced. However, during scale-up of plasmid-based systems, all or parts of these plasmid can be lost. The plasmid instability is undesirable for commercial systems. To overcome this problem, researchers have developed protocols for integrating a clone gene into a chromosomal site of the host organism. Under these conditions, the gene is maintained stably as part of the DNA of the host organism. The introduction and expression of foreign DNA in a host organism often change the metabolism of the organism and thereby impair its normal functioning. This phenomenon is called the metabolic load. A variety of strategies have been developed to minimize the extent of the perturbations caused by metabolic load and at the same time optimize the yield of the target protein and the stability of the transformed shell. Proteins produced by gram-negative bacteria may be engineered so that they are secreted to either the periplasm or the external medium by including a sequence encoding a peptide that marks the protein for secretion. This generally facilitates the purification of the protein and ensures that its expression does not interfere with cellular functioning. Many bacterial cells used to produce recombinant proteins tend to bind to solid surfaces and form biofilms. In this state, bacteria are resistant to high levels of antibiotics and limited in the amount of foreign protein that they can produce. To remedy this, it is possible to genetically engineer bacteria that are unable to form biofilms. An ideal plasmid expression system might include a convenient multiple cloning site in these different reading frames in which to insert the target gene, a strong but regulatable promoter, a selectable marker to eliminate the large background of non-transformed cells, a replication region for plasmid maintenance, or a site for homologous recombination. For integration of the plasmid into the host chromosomal DNA, a secretory signal peptide, and a removable, cleavable fusion tag that facilitates protein stability and eventually purification. However, both target proteins and expression systems are quite diverse, and investigators tend to develop a set of conditions that optimizes the production of a specific protein in a particular host cell. The differences in detail notwithstanding, the same fundamental strategies may be used to create a variety of different expression systems. That's it with this chapter, and let's go in for the review question. Right, we have the review questions. Suggest several ways that the expression of a clone gene can be for optimal expression. What is LAC-Q and how is it used? Why is the plasmid that contains the strongest promoter not always the best expression vector? That was number 3. Number 4. What is the TAC promoter and how is it regulated? The P1 promoter is derived from E. coli bacteriophage lambda, which cannot infect other bacteria, yet it is sometimes used as part of a broad host range expression vector. Explain how the P1 promoter can be used to promote transcription in organisms other than E. coli. Sometimes the strategy for the expression of a target protein in a host organism involves synthesizing the protein as part of a fusion protein. Why is this approach useful? How is a fusion protein created? What are inclusion bodies and how can their formation be avoided? Why would you want to express a foreign protein on the surface of a bacterium or bacteriophage? How would you do this? How would you manipulate the DNA sequences upstream of a target gene to modulate expression of that gene? How would you avoid some of the problems associated with the limited amount of oxygen that is available to growing E. coli cells when a foreign protein is overproduced? Number 11. A specific target DNA to be integrated into the chromosomal DNA of the host organism can include 1. Only the target gene sequence, or 2. The entire plasmid, including the target sequence. Explain how each of these results might occur. What are the advantages or disadvantages of the plasmid vector becoming incorporated into the host chromosomal DNA? What factors are responsible for a metabolic load? Suggest a number of different stages to limit the extent of a metabolic load on E. coli cells that are designed to overproduce a recombinant protein. During the course of integrating a target gene into the chromosomal DNA, the host bacterium, a marker gene may also be inserted to the chromosomal DNA. What strategy could be used to excise only the marker gene? How do biofilms limit the production of recombinant proteins? How might this limitation be overcome? How can E. coli host cells be engineered so that complex proteins with a large number of disulfide bonds are properly folded and therefore produced in an active form rather than as part of an inclusion body? So that winds up this entire lecture on chapter 6, which deals with manipulation of gene expression in prokaryotes. And we'll go into the next chapter shortly. Take notes and use them to the best of your capability and efficiencies and capacities for the examinations to pass the exams. And meanwhile, once again, I reiterate, please do not copy these any of these lectures for any commercial purposes. And let's give all due credits to the authors and those who have been more actively involved in making this book a success. Thank you. Best wishes. I'll see you in the next lecture. The continuing questions are: How can E. coli host cells be engineered to yield high levels of expression of foreign proteins that contain significant numbers of rare E. coli codons? This is a continuation to Chapter 6 review questions. Number 18 would be, what is the T17 expression system and how does it work? What features affect the strength of a bacterial promoter is number 19. The 20 would be, how can a protein of interest be engineered to be secreted to the medium by E. coli? And what is an inactin and how is it used to purify foreign proteins expressed in E. coli? So that winds up the review questions for Chapter 6. Welcome back. We enter Chapter 7 of Molecular Biotechnology, titled Heterologous Protein Production in Eukaryotic Cells. As we start on, we head on with the details of the subject. Now, the topics underlying this particular chapter are post-translational modification of eukaryotes and proteins, that's eukaryotic proteins, general features of eukaryotic expression systems, fungus-based expression systems, baculovirus insert cell expression systems, mammalian cell expression systems, summary, and the review questions follow. So for the first part, post-translational modification of eukaryotic proteins, an intro to that, where we have heterologous recombinant proteins from cloned DNA originating from a wide variety of organisms have been successfully produced using prokaryotic expression systems, although expression of any gene from any source organism in a prokaryotic host is theoretically possible. In practice, the eukaryotic proteins produced in bacteria do not always have the desired biological activity or stability. In addition, despite careful purification procedures, bacterial compounds that are toxic or that cause a rise in body temperature in humans and animals, paragens, may contaminate the final product. To avoid these problems, investigators have developed eukaryotic expression systems in fungal, insect, and mammalian cells for the production of uncontaminated therapeutic agents for either humans or animals. Large quantities of stable, biologically active proteins for biochemicals, biophysical and structural studies, and proteins for industrial processes. Moreover, any human protein intended for medical use must be identical to the natural protein in all its varieties. The inability of prokaryotic organisms to produce authentic versions of eukaryotic proteins is, for the most part, due to improper post-translational protein processing, including improper protein cleavage and folding, and to the absence of appropriate mechanisms that add chemical groups to specific amino acid receptor sites. All right, that's the intro, and on to the first part of it, the subdivisions. Post-translational modification of eukaryotic proteins. In prokaryotes, the steps in protein synthesis are not compartmentalized, and therefore translation of messenger RNA, mRNA, occurs concurrently with transcription. As soon as the nascent transcript emerges from RNA polymerase, it is accessible to the ribosome to begin translation. With the aid of folding proteins known as chaperones that bind to polypeptides as they are being synthesized, proteins are folded into their proper three-dimensional configurations during synthesis. In contrast, eukaryotes transport mRNA from the nucleus to ribosomes in the cytoplasm or in the endoplasmic reticulum, where translation occurs. Proteins produced on ribosomes associated with the endoplasmic reticulum either are inserted. Figure 7.1 is about the cleavage of inactive pro-insulin to yield active mature insulin. Proteases remove the leader peptide L and an internal peptide C, yielding a peptide that consists of chains A and B. Now to the subject, the membrane of the endoplasmic reticulum are secreted into the lumen of the endoplasmic reticulum during synthesis, where they are processed further. Right? Many proteins including those that are of interest as therapeutic agents for the treatment of human or animal diseases. There is some type of post-translational processing that is required for protein activity and stability. Some proteins are produced as inactive precursor polypeptides that must be cleaved by proteases at specific sites to produce the active form of the protein. For example, the small peptide hormone insulin is produced in animal pancreatic cells as a single polypeptide pre-proinsulin that is cleaved to produce two shorter peptides that are joined by disulfide bonds. Production of inactive pre-proinsulin ensures that the peptide is not active in the pancreatic cells that produce it, but upon active secretion cleaved mature insulin can act on other cells. Similarly, the digestive enzyme trypsinogen, which degrades proteins, is produced as an inactive polypeptide to avoiding digestion of components of the producing cell. Upon secretion into the small intestines, trypsinogen is cleaved by an entropeptidase to yield the active enzyme trypsin. Similar to prokaryotes, proper folding of proteins in eukaryotic cells requires the assistance of chaperones. In the endoplasmic reticulum, the chaperones BIP and calnexin bind nation polypeptides, and protein disulfide isomerases catalyze the formation of disulfide bonds between adjacent cysteine residues. Proper folding is important not only for the protein to attain a configuration for optimal activity, but also to protect regions of the protein that would otherwise be recognized by proteases that destroy the protein. Quality control systems ensure that only correct folded proteins are released from the endoplasmic reticulum and transported within vesicles to the Golgi apparatus for further processing. Proteins intended for secretion from the cell are subsequently transported to the cell membrane within specific transport vesicles and released by exocytosis. The addition of specific sugars, glycosylation, to certain amino acids is a major modification that provides stability and distinctive binding. Properties to a protein. Proper protein glycosylation is important because it contributes to protein conformation by influencing protein folding, can target the protein to a particular location, for example, through interaction with a specific receptor molecule, or an increased protein stability by protecting it from proteases. In the cell, oligosaccharides are attached to newly synthesized proteins in the endoplasmic reticulum and in the Golgi apparatus by specific enzymes known as glycosylases and glycosyltransferases. The different tissues may differentially glycosylate the same protein, thereby increasing protein heterogeneity because different sugar modifications can alter the properties of a protein. This presents opportunities for protein engineering to improve the efficacy or to alter the activity of a protein. Therapeutic proteins that require glycosylation for activity include antibodies, blood factors, some interferons, and some hormones. The most common glycosylation entail the attachment of specific sugars to the hydroxyl group of either serine or threonine, O-linked glycosylation, and to the amide group of asparagine, N-linked glycosylation. About 50% of all human proteins is glycosylated. The initial coarse sugar groups that are added to these amino acid acceptor sites tend to be similar among eukaryotes, although the subsequent elaborations among yeast, insects, and mammals are quite diverse, especially the N-linked glycosylation. Other amino acid modifications include phosphorylation, acetylation, sulfation, acylation, alpha-carboxylation, and the addition of C14 and C16 fatty acids, that is myristilation or myristilation and palmistylation or palmitylation, respectively. And unfortunately, there is no universally effective eukaryotic host cell that performs the correct modifications on every protein. In some cases, a host cell may add unusual sugars to either authentic or spurious amino acid sites and consequently create an extremely antigenic protein or possibly one that lacks its proper function. However, even though a recombinant protein may fall short of the stringent properties that are required for a therapeutic agent, it may still be useful for either research or industrial purposes. Different eukaryotic expression systems must be tested to determine which one synthesizes the largest amount of a functional recombinant protein. The choice of an expression system depends primarily on the quality of the recombinant protein that is produced, but the yield of produced product, ease of use, and cost of production and purification are also important considerations. General features of eukaryotic expression systems. The basic requirement of expression of a target protein in a eukaryotic host are similar to those required in prokaryotes. Vectors into which the target gene is cloned for delivery into the host cell can be specialized. Plasmids designed to be maintained in the eukaryotic host, such as the yeast 2 microarm plasmid, host-specific viruses, such as the insect baculovirus, or artificial chromosomes, such as the yeast artificial chromosome YAC. The vector must have a eukaryotic promoter that drives the transcription of the cloned gene of interest, eukaryotic transcriptional and translational stop signals, a sequence that enables polyadenylation of the mRNA, and a selectable eukaryotic marker gene. Because recombinant DNA procedures are technically difficult to carry out with eukaryotic cells, most eukaryotic vectors are shuttle vectors with 2 origins of replication and selectable marker genes, one set functions in the bacterium E. coli. Figure 7.3. Examples of some N-linked oligosaccharides in yeast: A, insects; B, and mammals. 3. All N-linked glycosylations in eukaryotes start with the same initial group, which is subsequently trimmed and then elaborated in diverse ways within these series and others among species. Some yeast sites have 15 or fewer mannose units co-series, and others have more outer chain family. In S. cerevisiae, the chains are frequently 50 or more mannose units. In asparagine N, residue next to any amino acid X followed by either threonine D or serine S can be targeted for glycosylation. Red circles, mannose; dark blue squares, N-acetylglucosamine; yellow triangles, glucose; green squares, galactose; orange squares, sialic acid; maroon triangle, and fucose. Another set functions of the eukaryotic host cell. If a eukaryotic expression vector is to be used as a plasmid, that is, as an extra chromosomal replicated DNA, then it must have a eukaryotic origin of replication. Alternatively, if the vector is designed for stable integration into the host chromosomal DNA, then it must have a sequence that is complementary. A segment of host complementary DNA, let's just have a look at that. To a segment complementary to a segment of host chromosomal DNA to facilitate insertion into a chromosomal site. The introduction of DNA into bacterial and yeast cells is called transformation. In these systems, the term describes an inherited change due to acquisition of exogenous foreign DNA. However, in animal cells, transformation refers to changes in the growth properties of cells in culture after they become cancerous. To avoid confusion, the word transfection has been chosen to denote inherited changes in animal cells that are due to the addition of exogenous DNA. The 3 techniques are commonly used to transform yeast: electroporation, lithium acetate treatment, and cell wall removal, protoplast formation. Transfection of cultured animals is achieved by incubating cells with DNA that have been co-precipitated with either calcium phosphate or diethylaminoethyl DAE dextran, or by electroporation entails subjecting cells to short pulses of electric current, thus creating transient pores through which DNA enters the cell. And viruses, lipid DNA complexes, and protein DNA aggregates are also used to transfer exogenous DNA into a recipient animal cell. Fungus-based expression systems. Fungi share many of the molecular, genetic, and biochemical features of other high eukaryotes and are therefore a good choice for heterologous protein production. Their growth advantages similar to those of prokaryotes, such as rapid growth in low-cost medium, generally do not require growth factors to be added to the growth medium, can correctly process eukaryotic proteins, can secrete large amounts of heterologous proteins. Initially, the yeast Saccharomyces cerevisiae was used extensively as a host cell for the expression of cloned eukaryotic genes. It has a long history of use in traditional biotechnologies in the brewing and baking industry. Today, a variety of fungal expression systems are available, and they have been optimized for recombinant protein expression. Versatile expression vectors with broad host ranges have been constructed because the optimal host for production of a particular target protein must often be determined experimentally in a number of different systems. Saccharomyces cerevisiae expression systems. High levels of recombinant protein production have been achieved using S. cerevisiae. The advantages of using the single-cell yeast are several. First, a great deal is known about the biochemistry, genetics, and cell biology of the fungus. The genome sequence of S. cerevisiae was completed in 1996, and it is used extensively in the study as a model organism for cell function. It can be grown rapidly to high cell densities on relatively simple media in both small culture vessels and large-scale bioreactors. Second, several strong promoters have been isolated from the yeast and characterized a naturally occurring plasmid called the 2 micrometer plasmid. Can be used as a part of endogenous yeast expressions vector system. Third, S. cerevisiae is capable of carrying out many translational modifications. Fourth, the yeast normally secretes a few proteins that when it is engineered for extracellular release of a recombinant protein, the product can be purified. Fifth, because of its years of use in the baking and brewing industry, S. cerevisiae has been listed by the U.S. Food and Drug Administration as a generally recognized as safe organism. It does not harbor human pathogens or produce fever-stimulating virogens. Therefore, the use of the organism for the production of human therapeutic agents, drugs, or pharmaceuticals does not require the same extensive experimentation demanded for unapproved host cells. A number of proteins that have been produced in S. cerevisiae are currently being used commercially as vaccines, pharmaceuticals, and diagnostic agents. Table 7.1, for example, at present more than 50% of the world's supply of insulin is produced by S. cerevisiae. Engineered S. cerevisiae strains are also major producers of hepatitis B vaccine. S. cerevisiae vectors. There are three main classes of S. cerevisiae expression vectors: episomal or plasmid, vectors yeast episomal plasmids, YEPSL, integrating vectors yeast integrating plasmids, YEPs, and YECs. Of these, episomal vectors have been used extensively for the production of either intra or extracellular heterologous proteins. Typically, the vectors contain features that allow them to function both as bacteria and as cerevisiae. An E. coli origin of replication and bacterial antibiotic resistance genes are usually included on the vector, enabling all manipulation to be first be performed in E. coli before the vector is transferred to a cerevisiae for expression. The YEP vectors are based on the high copy number 2 micro-M plasmid, the small circular plasmid found in most natural strains of S. cerevisiae. The vector replicates independently of the host chromosomes via a single origin of replication, autonomous replicating sequence, AR at STB loci, and is maintained in more than 30 copies per cell. Many S. cerevisiae selections can be seen. The schemes rely on mutant host strains. That require particular amino acid histidine, tryptophan, or leucine. Or nucleotide uracil for growth. Such strains are said to be oxytrophic because minimal growth medium must be supplemented with a specific nutrient. In practice, the vector is equipped with a functional wild-type version of a gene that complements the mutated gene in the host strain. For example, when a YEP with a wild-type LU2 gene is transformed into a mutant LU2 host cell and plated on the medium that lacks leucine, only cells that carry the plasmid will grow. The number of promoters derived from the cerebrosome genes available for producing engineering efficient transcription of heterologous genes and yeast vectors. Generally, tightly recolatable induced promoters are preferred for producing large amounts of recombinant protein at a specific time during large-scale growth. In this context, the galactose-regulated promoters respond rapidly to the addition of galactose with a 1,000-fold increase in transcription. Repressible, constitutive, and hybrid promoters that combine. The features of different promoters are also available. Maximal expressions depend on efficient termination of transcription. Often for YEP vectors, the terminated sequence is from the same gene as the promoter. Many heterogeneous genes are provided with a DNA coding sequence for an amino acid segment. Signal sequence, signal peptide, or leader sequence that facilitates the passage of the recombinant protein through cell membranes and its release to the external environment. The main reason for this modification is that it is much easier to purify a secreted protein than one from a cell lysate. The most commonly used signal sequence of S. cerebrosome is derived from the mating factor alpha gene. Also, synthetic leader sequences have been created to increase the amount of secreted protein. Other sequences that stabilize the recombinant protein protected from the proteolytic degradation and provide a specific amino acid sequence affinity tag that is used. And these extra amino acid sequences are equipped with a protease cleavage site so that they can be removed from the recombined protein after it is purified. Plasmid-based yeast expression systems are often unstable under large scale greater than or equal to 10 liters growth conditions even in the presence of selection pressure. To remedy this problem, heterologous gene is integrated into the host genome to provide a more reliable production system. A selectable marker gene into an S cerebrosome chromosome. Briefly, a functional selectable marker gene and a heterologous gene equipped with a yeast-specific transcription and a translation control sequence are inserted between two DNA segments. And a functional selectable marker gene, a heterologous gene equipped with a yeast-specific transcription and translation control sequences inserted between two DNA segments derived from the ends of a non-essential yeast gene. In this instance, the plasmid does not usually carry an origin of replication that functions in yeast cells. The plasmid is cleaved and the linear fragment is transformed into S. cerebrosome, a double recombination event between homologous sequences on the linearized plasmid, and a chromosome in the host inserts a piece of DNA with both target marker genes into a specific order. To a specific chromosome site, 7.5 is the figure. Plasmid DNA is linearized because DNA in this form is more likely than circular DNA to recombine with chromosome DNA. The DNA that is not integrated is lost during successive cell divisions. The major drawback of this strategy is the low yield recombination component protein from a single gene copy. Going back to the diagram, figure 7.5 is a schematic representation of integration of DNA with a YLP vector. A selectable marker gene And the gene of interest, GOI, with transcription and translation control elements not shown are inserted into a YLP vector between two segments from the ends of a non-essential yeast gene. The ampicillin-resistant AMP gene and the origin of replication ORI function in E. coli, a leucine requiring LU2 yeast strain is transformed with restriction content nuclease digested vector DNA because chromosomal DNA is more likely to recombine with linearized DNA than with circular DNA. Restriction endonucleosides flank the segments of the non-essential gene. The DNA sequences at the ends of non-essential gene A undergo recombination that leads to the incorporation of both the gene of interest and LU2 gene into the corresponding chromosome site. Transformers grow on medium that is not supplemented with leucine. Non-recombined DNA is degraded. This is